Alright, so this is our DNA model. This is in all its awesome completeness. And as you can see, it's a double helix structure. So it's like a bendy ladder.
Yeah, it's like a ladder with these rungs, which are the bases, and it's twisted like so, which is what makes the double helix structure. And so basically, the marshmallow, the base in the middle, and the blue Twizzler make up a nucleotide, and for your convenience, I've made a picture like so. So basically, the marshmallow is the sugar, and the blue Twizzler is the phosphate, and the base in the middle, yeah, so that attaches to the sugar, which attaches to the phosphate.
And so we have this really pretty key for you, so that the green Twizzlers represent the thymines, the purple represents adenine, orange represents cytosine, red represents guanine, and blue represents uracil, which is in RNA, which is, will be seen later, not here. Okay, so then in order to start the replication.
Okay, so in addition, these bases, adenine, guanine, cytosine, thymine, they're all different purines or pyrimidines, and the purines are represented by adenine and guanine, and the pyrimidines, which only have one carbon ring, are cytosine and thymine. So yeah.
Okay, so in order to start replicating, a helicase has to come over, and that's an avocado. So that's our helicase. So then it goes along the rungs and it breaks them apart. It breaks apart the hydrogen bonds. It's like, bam, they're all broken.
Yeah.
Okay.
And this is the next structure that we'll move on to.
And
so yeah.
This is the leading strand on your left side, and this is the lagging strand. And on the leading strand, this guy, which is a PEEP, it's the polymerase 3. It comes along and finds, it goes along this way because it's 5:3 ratio. It comes along and finds spare complementary nucleotides, which are just kind of floating around from the food you eat. So it goes on and adds them on as the ladder gets split by the helicase.
Okay, so this is called the lagging strand because this, instead of going from 5' to 3', instead it also goes 5' to 3', but because the carbons are basically upside down, going 5' to 3' means going this way. So what happens is the RNA primase, which is represented by this tomato, what it does it lays down an RNA primer, which is like RNA complementary nucleotides. So as the helicase continues to break, the RNA primer comes down, and then the RNA primase lays down another section of RNA primer, and then the DNA polymerase 3 realizes, oh, it's time to do my job. So it goes down, it starts at the beginning, it starts at the bottom of the first section of RNA primer, and then it lays down the DNA complementary nucleotides until it reaches the top of the second section of RNA primer.
And then once that's all done, the polymerase 1 comes and it essentially replaces the RNA primers with DNA. So essentially, these blue thingies would turn, these yellow thingies would turn into blue thingies. So it does that for this part, skips over this part because it's already DNA, and then it does it for this part. And right now they're all in Okazaki fragments.
So this DNA ligase, which is represented by this cherry tomato, connects these Okazaki fragments so that the entire strand becomes a whole. And also this is called semi-conservative replication because the new strands of DNA, they each have one strand of the original DNA and one strand of the new DNA. Thank you. Yeah.